Do you remember what we have discussed in earlier class? We discussed about how to identify the bacteria. This is just a continuation of earlier class. In earlier class, we discussed about why we need to identify the organism and how we are identifying colony characteristic and biochemicals that we have discussed in earlier class. And today we'll go in depth about biochemicals. Then again, identification of bacteria will not be complete. So later on, we'll be talking individually on different bacteria that you are going to practice for. So I would like to continue the class with how we approach for the identification of the bacteria. In the examination, during the class, you will be taught about these 5 organisms: Staphylococcus aureus, Pseudomonas aeruginosa, Proteus mirabilis or Proteus vulgaris, anything it could be, Escherichia coli, Klebsiella pneumoniae. Here among these 5, one is gram-positive cocci in cluster, rest all are gram-negative bacilli, and these all are common pathogen that we encounter during the clinical practice. That's why they are kept here for identification. And today we are not going to talk about individual bacteria, but we are going to talk about only those chemicals that we use for identification of these bacteria. It's just a recap of earlier class. For the steps of identification of organism, the first and foremost is identification of medium, because in different medium, different organism, or in a different medium, organism may have different colony characteristic. So whenever we are seeing an organism in a medium, first thing in our mind should be, what is the medium? What colony characteristic of bacteria can have in this medium? Like if we are seeing a blood agar, at the time we can see hemolytic property of an organism in a blood agar can be seen over there. That's why organism, when seeing in a blood agar, we need to think whether it is hemolytic or non-hemolytic. If it is hemolytic, what type of hemolysis? It is beta hemolytic or alpha hemolytic. That we need to think. Similarly, if we are using McConkie agar, then there lactose is there, indicator is there. If some organism is lactose fermenter, then it will make pink colonies. If some organism is there and that is not lactose fermenter, then they will make pink pale colonies or yellowish colonies. So while seeing the organism, first we need to think is, what is that media? According to that, we can see the useful property of bacteria. Similarly, there can be slight differences in colony characteristic based on medium also. At this point you can forget. Just remember, certain characteristics are different in different media. That's why we need to identify the media. Then colony characteristic. In earlier class, if you remember, we have seen some colonies are round, some colonies having regular margin, some colonies are not having regular margin. Some bacteria are producing colonies with rough surface, someone with glistening surface, someone with a wrinkled surface, someone are making colony more than 5 mm in diameter, some are making very tiny colonies. That kind of differences we have seen, and that differences are that colony properties or characteristic is unique for different organism. That's why colony characteristic features in a particular media are add-ons property, or they can be cumulatively seen to identify the organism. After that, what we do is gram stain. Along with gram stain, I told you we may do Zn staining also. We may do other special staining also. Gram staining is almost for all the organisms we do. If we are suspecting organism, that could be mycobacteria. If we are seeing the organism that is producing the spore, at the time we may do ZN staining or modified ZN staining based on need. So suspicion is there, then based on it we are doing gram stain, we are doing ZN stain, or we are doing certain special staining. If we suspect Corynebacterium diphtheria, then we go for Albert stain. So after this staining, now after staining is over, what we do? Sometime organism will come gram positive, sometime organism will come gram negative. Sometime organism may not get stained at all. So there are very less organisms that won't get stained by gram stain. Now it's your job to find out what are the organism or list the organism that will not stain in gram stain. It is your job. So now we'll forget about not staining, that don't get stained by gram stain. That's your job to find out and present to me. Now in gram stain we get either gram positive or gram negative. So gram positive we go for particular set of gram biochemical properties, while in gram negative we'll go for particular set of biochemical properties. So these sets are, you can see here, triple sugar iron test. We'll come to this test later. Now remember the name only. Triple sugar iron, TSI, methyl red, VP, Vox Proscar, Simon's citrate, Christensen's urease, sulfide indole motility or SIM, and oxidase, catalase, coagulase. So these are the different tests we do. Beyond this also plenty of tests are there, but at least for you this test you have to do, you have to interpret. That's why do not forget. So remember these biochemical properties. These all are used for those organisms that we have already listed that you are going to identifying, and that is one of the important practical examination part also. One of the organism is given to you in the practical. You have to do or interpret all these tests and identify that organism. So now remember this name: TSI, MR, BP, Simon's citrate, Christensen urease, SIM, oxidase, catalase, coagulase. Now after this what? After this we see all the properties, all the biochemical features, and we come to conclusion that this organism is this, or this organism can be E. coli, this organism can be Klebsiella, this organism can be Staph aureus. This is the how we identify the organism. Same in the laboratory, same here for your practical. These are the same tests that we have already listed. Now more important is this one. What is the basis of those tests? What are the basis of those biochemical reactions that we have already listed as the name? These biochemicals are, certain biochemicals will contain sugar, and with the use of that biochemical we inoculate organism there, then we'll see how the sugar is being utilized, whether they are fermentatively utilized or oxidatively utilized, or they don't utilize at all. Like certain organism can ferment glucose, certain organism cannot ferment glucose, but they can utilize glucose in presence of oxygen, and certain organism cannot ferment, cannot utilize any of the sugar, neither monosaccharide, neither disaccharide. Any of the sugar they cannot utilize at all. And those organisms are called saccharolytic. Saccharolytic. It came from the word saccharolytic. Saccharolytic means saccharose sugar. Lytic means that can use it, split it. A when it comes before, then it gives the negative meaning. They cannot at all. So this is one of the how we test utilization of sugar. Same with amino acid. In a media, we use a particular type of amino acid and see whether that amino acid is used or not. Similarly, their ability to use citrate as a source of carbon. Now, here you have to remember why we need the carbon. What happens if we don't have any carbon at all? Now think biochemistry. What are our biomolecules made up of? Almost all they have carbon. If we see fermentation, if we see TCA cycle, sugar goes a particular cycle, and from their byproduct will be utilized in different other cycles. And these all biochemicals are made up of carbon and something other. Same with the bacteria. Carbon is needed everywhere. Almost all biochemical molecules, biomolecules, bigger molecules, they are having carbon. So carbon is needed anyhow, survival. And from where the carbon will come? Carbon may have different sources. Glucose is one of the source. During going fermentation cycle, in between byproducts are formed. They are the source of carbon. They are utilized for some other cycle. They are the source as a source of carbon. Sometime CO2, carbon dioxide, can be source of carbon. Acetate can be source of carbon. Methanol can be source of carbon for the bacteria. Similarly, one of the source is citrate, which is the source of the carbon for bacteria. But all the bacteria cannot utilize citrate. That means in a media, if we can use citrate alone, only citrate as a source of carbon, no other molecules that contains carbon is there. Then if some bacteria survives on that media, then citrate is utilized. If bacteria cannot survive on that media, then citrate is not utilized as a source of carbon. So that is one of the tests. Similarly, we can look for metabolic pathway of glucose fermentation. Bacteria, if we see the how glucose is being fermented, what is the pathway? After fermentation is complete, whether it will lead to the final product with very low acidity or final product with high acidity, or acidity at below 3 or around 5 to 6. Different end products of the glucose fermentation that we can test. Certain bacteria will ferment the glucose and make acidity very low, and that it can keep for a long period of time. Certain bacteria, they will ferment the sugar, lactic acid is formed, but acidity because of that will not last for a long period, and final acidity of that medium becomes higher. It will be acidic, but it will be higher. So that is the different pathway. That glycolytic pathway, we can see the different details of it. We can come later. We'll discuss later. So just now remember, pathway of glucose metabolism is different. One will lead to low pH, low and stable pH. Another will lead to high pH. That can be identified. And this is different in different bacteria. Now, production of different enzymes, ureas. Urease, if produced by bacteria, that is able to uh, spread the urea. Catalase can spread the H2O2. Oxidase, tryptophanase. Just now remember, these are the different enzymes that we are utilizing in our test that we have already been listed, and that can be checked in the biochemical test. Virulence factor, coagulase for certain bacteria, or better say Staphylococcus aureus. We can test for. If it is found to be producing coagulase, then it is Staph aureus. If not, some other than Staph aureus. Staphylococcus only, but some other than Staph aureus. Motility, whether a bacteria is motile or not. Gas production, or end product of fermentation. At the end, fermentation will lead to gas production or not. Usually after fermentation, gas is produced, but certain bacteria, their fermentation will not lead to production of gas. H2S production, whether they are able to produce H2S or not. These all tests are for these all biochemical properties we can design a test to check for. And those tests we have already listed, different tests already listed earlier, we'll see again. So these all tests can be features of the bacteria can be detected, and this will lead to definitive identification of a particular bacteria. Now you have to understand indicator. Indicator are the chemicals, substances, which will change color due to change in pH. If we see these all tests here, these all activity will lead to either acidic pH or basic pH. And so that pH changes will be detected by indicator. Different indicator can be methyl red, phenol red, bromothymol blue. Same with in culture media. In McConkey agar, we use certain indicator. In TCBS, certain indicator will be there. So these indicators are same. They have different color at acidity, different color with change in or shift in acidity. Or in basic pH, different color, acidic pH, different color, neutral pH, different color. These are the indicator, and this indicator will be incorporated in a medium, and bacterial activity can be detected. This is the biochemical test that we have already listed. We'll just see the name, triple sugar iron test. This means it has 3 types of sugar, so we can see the fermentation pattern. Methyl red and Bose-Prosker can come together. If you remember, this will detect the metabolic pathway of glucose fermentation. Simon citrate. If you remember, in there one of the point was citrate utilization as the source of carbon. So this is test for citrate utilization. Christensen's urease. This is one of the tests for enzyme production, that is the enzyme urease. Sulfide indole motility, H2S production, indole production, but better to understand here whether the organism is having tryptophanase or not. If organism is producing enzyme tryptophanase, then indole will be produced, otherwise not. So indole is a test for enzyme production. Motility, whether the organism is motile or not. Catalase, a test for enzyme catalase, whether produced or not. Coagulase is a virulence factor, whether the organism is producing or not. Similarly, oxidase is an enzyme, whether it is actually test for cytochrome oxidase, whether it is there or not. So these are the different tests that we utilize during if we utilize here for the identification of organism. Slowly we'll come to this point. Now see, this is the triple sugar iron agar. The use of triple sugar iron agar is we can check for carbohydrate fermentation pattern. It contains 3 sugar: lactose, sucrose, and glucose. So whether lactose is fermented or not fermented, glucose fermented, not fermented, or glucose only fermented, that we can identify. So it gives the carbohydrate fermentation pattern. Gas production. If organism is fermenting this sugar and one of the end product is gas, and that can be detected in the medium. We'll see how it is detected. So gas production and hydrogen sulfide production. It will contain certain chemicals that will trap the hydrogen sulfide and convert into ferrous sulfide, which is a black precipitate. So it can be used for hydrogen sulfide production test also. Now what is the content of the media? As name suggests, it contains 3 different sugar: lactose, sucrose, and glucose. Concentration wise, lactose and sucrose is 1% and glucose is 10 times lesser, 0.1%. That means if 10 part is lactose, 10 part is sucrose, then 1 part is glucose. Or 10 gram is lactose, 10 gram is sucrose, then 1 gram is glucose. The advantage of making this is if organism is saccharolytic, fermentative, then their first target will be glucose. Then only they will go for lactose and sucrose. So glucose will be depleted first. So fermentation of glucose can be easily detected. We'll see later how this glucose fermentation alone is detected. Our glucose and sucrose fermentation is detected, or all 3 sugars are detected. We'll see the picture later. Along with lactose, it will these sugars, it will contain other regular nutrient. That means amino acids will be there, peptones will be there, these all will be there. And here indicator used is phenol red. So in basic or neutral pH, it will be orange red in color. In basic pH, it will be purple in color, and it will change to yellow in acidic pH. So if sugar is fermented, it becomes yellow. If sugar is not fermented, then it will either remain as a same color, or it may convert into purple color. What is happening here is bacteria that is not able to utilize any of the sugar has to survive, and it will utilize peptone or amino acid. The end product of utilization of amino acid is basic. That's why those organisms that will utilize amino acid will make basic environment. That's why the phenol will convert into purple in color. Now see here. I'll just change my pointer. To utilize amino acid, there must be presence of oxygen. That means only in surface it is possible to utilize amino acid. So only in surface, in this area, amino acid will be utilized. Only this area will be basic in pH. That means for those organisms that are unable to utilize sugar, will convert only this surface purple in color. Now down to it will be no change at all. This is for the organism that is not able to utilize the sugar. Certain organisms are like that, which will utilize only glucose. Now, what is happening for only glucose is, first they will utilize glucose. They will change the pH. Acidic pH will be maintained. Whole media will be converted into yellow in color. Now, glucose is just 0.1%. So, glucose will be depleted within 6-7 hours. After that, they have to survive. Then they will start utilizing the amino acid. So, till glucose is depleted, everything is yellow. But after this, bacteria started using amino acid. But that is possible only in the surface. So, after utilization of amino acid, this area becomes basic in nature. So, phenol red, because of phenol red, this area will convert into purple, and below there will be yellowish in color. We'll see the picture later. Now remember, those organisms that are able to utilize sugar only will be having this area yellow and this area purple in color. Now for those organisms that are able to utilize more than one sugar, glucose plus sucrose or glucose plus lactose or glucose plus sucrose plus lactose, the finding will be same. The finding will be acidic pH only throughout the media. So at the time, whole media will be yellowish in color. And if they are producing gas, then gas can, since this is solid media, gas cannot escape. So they will make the space like this. There will be cracks, there will be upliftment of media, and we can see the gas like this. Now for those organisms that produce hydrogen sulfide, then that hydrogen sulfide will be trapped by ferrous ammonium sulfate, and ferrous sulfide will be formed, and which are not a gas. Ferrous sulfate is not a gas, so that will be a precipitate seen like this. If they are heavy H2S producer, then whole thing will be black. We'll see the picture later. Or they are very low H2S producer, then very less area will be black. This is the color of TSI. Now we have discussed plenty of things about TSI. Now while interpretation, I have told you, in some bacteria this area will be purple, some bacteria this area will be yellow, some bacteria whole thing will be yellow. So here you have to understand, for TSI, for interpretation of TSI, we see two things. One is this slant, this slope, half of this slope, other is this butt area. So whenever we are saying interpreting the TSI, say in terms of slope and in terms of butt area. We'll see later. If you remember, this happens if organism is able to ferment the glucose only. After that, it will start utilizing the amino acid, but the amino acid utilization is possible only in presence of oxygen. So only in the slant amino acid will be utilized. That's why only this area is getting purple in color. Below that, because of sugar fermentation, glucose fermentation, it is yellowish in color. This is the condition when bacteria is utilizing more than one sugar. Glucose plus sucrose or glucose plus lactose or glucose plus sucrose plus lactose. And there is no production of gas. At the time, slant and both will be yellow by yellow. This one we can say purple by yellow. This is the condition where bacteria is utilizing at least 2 sugar. As said earlier, glucose plus sucrose or glucose plus lactose or glucose plus sucrose or lactose with production of gas. Compare A and B. Here crackles will be there, is there. Media is lifted up, but slope as well as butt is yellow in color. So it is yellow by yellow. Gas production is positive. Here yellow by yellow gas production is negative. This is the condition when there is heavy production of H2S. And this slant is here amino acid is being utilized, and this area is purple, and below that heavy production of H2S is there, which will react with ammonium thiosulfate or ferrous ammonium thiosulfate, forming ferrous sulfide, which is a precipitate. This is the condition when some bacteria is completely asaccharolatic. At the time they will utilize on the surface only amino acid and convert the media color into purple. Uninucleated, both slant and butt orange in color. Shigella, here you can see slant is purple in color, butt is yellow in color. That means only glucose is being fermented. Same here, and here pseudomonas growth is there. Since they are asaccharolatic, they can utilize any of those three sugars. That's why it is purple in color, and there is less color change in butt. This is in case of E. coli. In case of this is in case of Proteus. This is in case of E. coli. This is not changed at all. For individual bacteria, now do not get confused. Just remember the test, different changes, and how to interpret. And what does that change in color mean? For an individual bacteria, again we'll be talking about these biochemicals. At the time we'll come to each and every examples for these different biochemical tests. So I am not saying you any of the example, any of the test positive or test negative here, because I don't want to make you confused. I just want today for you to remember the name of the test, and for you to remember how the test gets changes and what does that signify. Just remember that much. Later how to interpret, correlate with the organism identification will come again while talking about individual organism. MR BP test. Now MR and BP, they will come together. Methyl red and Box Proskar. If you remember earlier what I told you, some tests are designed to detect glucose fermentation pathway. A particular set of organism, I told you, will form low pH after fermentation and low as well as stable low pH. And certain organism will make acidic pH after fermentation, but that acidic pH will not be maintained very low and will remain as higher around 5 or around it pH. So these are the different metabolic pathway of sugar glucose fermentation. And a same organism cannot maintain both the pathway. So either the organism will make low and stable pH or make low pH but a pH acidic but comparatively high. and those different fermentation pathway are named as mixed acid fermentation pathway or 2,3-butanediol fermentation pathway. Here in mixed acid fermentation pathway, you can see plenty of sugars, acids are being made. That's why the pH will be very low, and that low pH is stable. And it is detected by methyl red test. And 2,3-butanediol fermentation pathway will make acidic pH but not very low. Instead, it will make comparatively high pH, high acidity will be maintained. So that is detected by VP test. Now, since we have seen two pathway that is going entirely in a different way, a same organism cannot be MR positive and VP positive together. At a time, they are either MR positive or VP positive. Or in other words, if organism is MR positive, that organism will definitely show VP negative. If that organism is VP positive, then that organism will definitely show MR negative. Details of test will come again later. Now, what I insist you to remember is methyl red MRBP test is detecting glucose fermentation pathway first. And in pathway, there are two pathways. One will lead to low and or stable low pH that is detected by MR test, and in another pathway will lead to acidic pH but comparatively high pH, and that is detected by VR test, BP test, and MR and BP can never be simultaneously positive at the same point of time for organism. And those pathway are called mixed acid fermentation pathway and 2,3-butanediol fermentation pathway. Just 5 sentences is right now good enough. This is the test, MR test. Media will contain glucose and buffer and peptone, but basic for survival and buffer. Organism will be cultured over there, and methyl red, since it is an indicator, will detect the pH. Just remember, positive is red in color, brick red in color, negative is yellowish in color. BP test, same, glucose phosphate broth is the media. If organism is positive, it is pink in color or purple or red in, not purple, red in color. If it is negative, it won't be red in color. There are different methods. Right now, in coming classes, we'll talk about method also. Right now, do not get burdened. Just think about MR and BP. What is their use and what is the change in color in positive and what does that signifies. That is good enough right now. Citrate utilization test. The media, we call it as a Simmons citrate based on the person who has devised the medium. In book, you might encounter with the Cozus citrate also. There is also test for citrate utilization, but before Simmons, plenty of demerits are there. If needed, will come later. Otherwise, just now remember Simmons citrate. It will detect an ability of an organism to utilize citrate as a sole source of carbon. So in this media, there is no any other source of carbon other than citrate. If organism can utilize the citrate for the source of carbon, then it can survive. If it cannot, then it can't survive. If it can survive and it is able to utilize other content, that is inorganic ammonium salt, at the time they will form ammonium hydroxide as a byproduct and make the medium basic. At the time bromothiamyl blue will convert into green to blue in color. So change in color is indication of organism has utilized citrate as a sole source of carbon. Sometime organism is able to utilize citrate but not able to utilize inorganic ammonium salt as a source of nitrogen. At the time only growth of organism will be seen but no change in color. So growth only or change in color is indication of positive citrate utilization test. So here what you have to remember is test is citrate utilization, medium Siemens citrate medium. In positive it is green in color, in negative it is blue in color, or growth will be visible. In negative there is no change, organism cannot grow on the media. This much you have to remember. After that, one more point you have to remember is if organism is surviving, they will utilize ammonium salt as source of nitrogen. That will lead to formation of ammonium hydroxide, will change to media basic, and that will be indicated by indicator bromothiamyl blue, which is changing the media from green to blue. This is the different media: uninucleated green in color, citrate negative green in color, citrate positive blue in color. Urease test. Certain bacteria will produce urease enzyme which can split urea into carbon dioxide, water and ammonia, and that ammonia will react with water to form ammonium hydroxide, a basic media, a basic environment. That means ammonium hydroxide can dissociate into NH4 and OH. So that means that will convert the media into basic environment. So there is phenol red or indicator, and that will convert the media color. So in Christensen's urea agar, what you have to remember is urea will be there, phenol red will be there, and other basic requirement for the survival of the organism will be there. If organism is able to split the urea, will convert carbon dioxide, water and ammonia. These two will react and form ammonium hydroxide, which is a weak base, will convert the media into basic, and that will be represented by phenol red converting into color yellowish to pink. So organism which is urease negative will be yellowish in color, which is urease positive will be pink in color. This is the urease test, a test for enzyme urease. This is the different tests you can see. All pink colored are urease positive, and urease negative that you can see here, yellowish in color, they are either urease negative or uninucleated, or original color is like that only. So it is the urease test. Here you have to remember it is the test for urease enzyme. It is the test for ability to split urea into carbon dioxide, water and ammonia. Ammonia will react with water, forming ammonium hydroxide, which makes medium basic, and that is represented by phenol red. Sulfide indole motility. This is the test for sulfide H2S production. Indole enzyme tryptophanase, and motility, whether the organism is motile or not. The medium we call them as a SIM agar, sulfide indole motility, that is the SIM. And here the three physics are checked: H2S production, indole production, and motility production. H2S production is just like H2S production mechanism in triple sugar iron agar. H2S will be produced, that will react with ferrous ammonium sulfate, and ferrous sulfide will be formed, which is a solid precipitate that will be trapped and seen as you see in the picture. And this motile media is semi-solid. That means if organism is motile, then that will give a diffuse growth. Organism from the stab line that will try to grow in a diffuse fashion. So if organism motile, diffuse growth will be seen. If not motile, growth will be seen only along the streak or stab line. Indole production, I told you earlier also. This is the test for enzyme tryptophanase. If organism is producing tryptophanase, that will be able to split or use tryptophan or degrade tryptophan, leading to indole production. Indole is a gas which will be detected by use of Kovacs reagent. As you see here in the picture, those organism that is having tryptophanase enzyme is converting into this Kovacs reagent into pink in color. Those which are not able to utilize tryptophan or they are not having tryptophan enzyme, then there will be no change, or we say it as indole negative, indole positive. So this is the sulfide indole motility test. Here you can see the first picture. Organism is been growing, trying to go away from the line of streak. So it is motile. On the other hand, you can see there is a growth of organism only along the streak. It's not diffused like this. That's why organism is non-motile here. Organism is motile here. Here is the indole produced, indole not produced. H2S produced, H2S not produced. H2L produced, H2S not produced. Sometime indole, instead of this strip, liquid will come, Kovacs reagent, and that liquid, if we pour over here on the top of the surface of the media, then it will change in color in case of indole positive, and no change in color in case of indole negative. See, this is the picture you have already seen. You can see indole produced, you can see H2S produced, you can see motile, you can see non-motile. This is the same picture. Indole production, not production, motile, non-motile. Here you can see motile organism, diffuse growth, non-motile growth only along the streak line. Oxidase enzyme. Oxidase production. Now at this point, I would like to ask you, do you have any confusion earlier test till now what we have discussed? Please speak up. Are you clear? It needs time to memorize all these tests, all the interpretation for all the organism that we are going to discuss at the time. This test will again come at the time. Individual test we can discuss again, go in detail further than what we haven't discussed. We can come. So till now, just tell me any queries you have or not. Someone please speak. Unmute your microphone and speak. Hello. Hello. Hello.
Yes, sir.
Okay. I know this is very much confusing for you. So Mm, different biochemical tests we have read. You go through it, make a note by your own. At the time you can write, find it, which are positive, which are negative, make note, and any queries, just jot it down and ask me uh, through these Microsoft Teams or in next class, it doesn't matter. Just feel free to ask. Then remaining tests like oxidase, catalase, coagulase will come in coming classes. Uh, okay, is it okay? Any queries you want to ask? Till now we have discussed up to 1, 2, 3, 4, 5, 6 tests only, and catalase, coagulase, and oxidase we'll discuss in coming class. Okay. Any queries?
No, sir.
Okay, I know it is very difficult for you. It will be difficult for you to remember also. So make a note, and any queries, just jot it down and ask me. So it's fine. We'll end today's class here. We won't make any more burden with this test. We'll come to discuss in coming class. If there is no queries, then it's good. Uh, have a good day. Bye bye.